Greetings everyone and welcome to my channel. In this video we are going to talk about DESeq2 package and how does it work. DESeq2 is a widely used R package for analyzing RNA-Seq data to detect differentially expressed genes. It is a powerful tool in the field of bioinformatics and provides robust statistical methods for identifying gene expression changes between different conditions or groups. RNA-Seq data is generated from next-generation sequencing experiments and produces row read counts for each gene in each sample. The data is represented as a count matrix, where rows correspond to genes and columns correspond to samples. Each cell in the matrix contains the counts of reads mapping to a specific gene in a particular sample. Normalization is a crucial step in RNA-Seq data analysis. It aims to remove biases due to variations in library size, RNA composition, and sequencing depth among different samples. Proper normalizations ensures that genes' expression levels can be accurately compared between samples. Let's dive into library size, RNA composition, and sequencing depth. For library size variation, library size, it refers to the total number of reads obtained from sequencing a sample. Library size can vary significantly between different samples, which can introduce bias in the RNA-Seq data. Larger libraries may have more reads mapped to each gene, leading to higher row counts for all genes. Let's go for an example to make it easier to understand. For example, suppose the library sizes for 3 samples are as follows. S1 or sample 1 has 50 million reads, while S2 60 million reads, and S3 70 million reads. In this case, the row counts are directly influenced by the library size. Note, when we perform RNA-Seq to measure the gene expression in different samples, the number of reads obtained for each gene is known as row counts. However, the total number of reads or the library size can vary between different samples. This happens because some samples may produce more reads during sequencing than others. For instance, gene G1 has higher counts in S3 compared to S1, simply because S3 has a larger library size. To address this, we need to normalize the data. Now imagine we have a gene, let's call it gene X, and we are comparing its row counts into 2 samples, S1 and S3. In S1, the library size is relatively small, 50 million reads for example, and the row count for gene X is 10 reads. In S3, the library size is larger, for example, 70 million reads, and the row count for gene X is 20 reads. Because the library size is larger in S3, it's expected to have more reads for all genes including gene X. Therefore, the row count for gene X in S3 is higher than in S1, not necessarily because gene X is more highly expressed in S3, but due to the larger number of reads produced by S3. Note, when we say that a gene is highly expressed in a specific sample, such as S3, it means that the gene is producing a larger number of RNA molecules, mRNA, in that particular sample. And now, the RNA composition variation. RNA composition, it refers to the portion of different RNA types, mRNA, rRNA, tRNA, in a sample. Variations in RNA composition can affect the efficacy of library preparation and sequencing, leading to differences in gene expression counts. For example, suppose samples S1 and S2 have a higher proportion of ribosomal RNA compared to S3. This higher rRNA content may result in fewer sequencing reads available for mRNA sequencing, leading to lower counts for mRNA transcript in S1 and S2 compared to S3. And sequencing depth variation. It refers to the number of reads mapped to each sample during sequencing. Unequal sequencing depths can lead to differences in the total number of reads obtained for each gene, resulting in differences in row counts. For example, suppose samples S1 and S2 were sequenced with a sequence depth of 50 million reads each, while sample 3 was sequenced with a depth of 30 million reads. Genes with higher expression levels may have more reads mapped to them, and the differences in sequencing depths can impact the row counts for each gene between samples. For example, in other words, imagine you have 3 boxes, box 1, box 2, and box 3. The number of balls or reads in each box tells us how active a gene is. Now we want to compare the activity of a gene between these boxes. But there is a problem. Box 1 and box 2 have bigger sizes and can hold up to 50 balls each. But box 3 is smaller and can only hold up to 30 balls. Since the boxes have different sizes, it's not fair to compare the number of balls directly. We need to adjust or normalize the counts to make it fair. DESeq2 helps us to do that. It takes the size differences into account and adjusts the counts, so we can compare the gene activity accurately. How DESeq2 performs statistical testing, negative binomial distribution, dispersion estimation, calculation of test statistics, statistical significance analysis. DESeq2 constructs a statistical model based on the negative binomial distribution for each gene. But why negative binomial distribution? For example, one of the simplest models for count data is the Poisson distribution, which assumes equal mean and variance. So why not Poisson distribution? However, in RNA-Seq data, we often encounter over-dispersion, where the variance is larger than the mean, and hence the variance is not equal to the mean. We can't use Poisson distribution. This occurs due to various biological and technical factors, such as variability in gene expression, library preparation, and sequencing efficiency. The Poisson distribution cannot capture this over-dispersion, leading to inaccurate estimation of variance and potentially incorrect identification of differentially expressed genes. Some might think of using the normal distribution for transformed RNA-Seq data. However, applying the normal distribution to row counts can lead to negative or fractional values, which are biologically meaningless. The negative binomial distribution comes to the rescue. DESeq2 uses a negative binomial distribution model which considers both over-dispersion and zero inflation. It's specifically designed for count data and can handle excess zeros and variance greater than the mean. The model includes the row count data and the normalization factors, which are the size factors, obtained during the normalization step. The size factors are used to scale the counts and make them comparable between samples, accounting for differences in library size. Negative binomial distribution, Kij, which means the row counts for gene I in sample J, MB, which is the negative binomial distribution, Sij, Qij, which is the mean is taken as normalized counts scaled by normalization factor, Sij is the sample size factor, and Qij is the expected true concentration of fragments for sample J. Alpha I or alpha sub I is the gene-specific dispersion parameter. The Sij Qij is the fitted mean, which is the sample size factor times the expected true concentration in sample J. Qij is the expected true concentration of fragments for sample J. A log 2 Qij equal to x sub j or xj times beta sub I or beta I. Log 2 Qij equal to xj times beta I. This explains how the expected counts Qij are calculated from the observed counts Kij after accounting for various factors, including library size and gene-specific effects. Parameter Qij. This parameter is proportional to the expected true concentration of gene fragments in sample J. In simpler terms, it quantifies how much of the gene's expression we expect to find in that sample. Log 2 Qij. This transformation is commonly used in RNA-Seq data analysis to stabilize the variance across the range of counts and make the data more suitable for statistical analysis. To further model the data, we use coefficients beta I associated with each gene I in the model matrix X. These coefficients give us the log2 fold changes for gene I in comparison to a reference condition or a group. They tell us how much the gene's expression level changes between conditions. In DESeq2, we use a mathematical model to understand how genes' expression level change between different conditions or groups in an experiment. This model includes something called coefficients, represented as beta I or beta sub I. Each coefficient beta I is like a special number associated with a specific gene. It tells us how much the gene's expression changes between two conditions or groups being compared. Here's how to interpret the coefficient for a gene. If the coefficient beta I is positive, the gene is upregulated, which means it becomes more active in one condition compared to another. If the coefficient beta I is negative, the gene is downregulated, which means it becomes less active in one condition compared to another. If the coefficient beta I is 0, there is no difference in gene expression between the two conditions. In the case of the beta I coefficient is positive, let's assume the same example but replace the conditions with cancer and normal. Cancer sample 1 J equal 1 has 10 reads. Cancer sample 2 J equal 2 has 15 reads, but normal sample 1 J1 has 5 reads, and normal sample 2 J2 has 8 reads. Step 1, we calculate the mean expression for each condition. The mean expression in cancer mu ij for j equal 1 and j equal 2 equal to 10 plus 15 over 2, which is 12.5. And the mean expression in normal equal to 5 plus 8 over 2, which is 6.5. Step 2, calculate the log2 fold change for the gene beta I. Beta I log2 to mean expression in cancer over mean expression in normal, which is log2 12.5 over 6.5, which is approximately 0.88. Since the log2 fold change beta I is positive 0.88, it means the gene is upregulated in cancer compared to normal. This suggests that the gene is more active in cancer, with approximately 0.88 times higher suppression compared to normal. Assume we have two conditions, control and treatment, and two biological replicates in each condition, total of 4 samples. Our design matrix X might look like this. We have intercept and condition treatment columns. Intercept 1, condition 0, intercept 1, condition 0, intercept 1, condition 1, and intercept 1, condition 1. In this case, we have 2 samples in each condition. We can represent the design matrix X as 4 times 2 matrix, where each row corresponds to a sample, and the columns represent the intercept and the condition, which is treatment. Now let's assume we are interested in a specific gene, gene I, and we want to calculate the log2 fold change beta I between the treatment and control conditions for this gene. Suppose the values for xj for each sample are xj for S1, which is control 1, 1 and 0, xj for S2, control 2, 1 and 0, xj for S3, treatment 1, 1 and 1, xj for S4, treatment 2, 1 and 1. For gene I, the log2 fold change beta I is estimated by fitting the DESeq2 model to the count data, accounting for the gene's specific dispersion, alpha I. The model will use the information from the design matrix X, including the values of xj and the observed counts, to estimate beta I. The log2 fold change beta I represents the change in gene expression between the treatment and control conditions. If beta I is positive, it means that the gene is upregulated in the treatment condition compared to the control condition. If beta I is negative, it means that the gene is downregulated in treatment condition compared to the control condition. The xj values in the design matrix X help incorporate information about the experimental conditions of each sample. In this example, the first column of X represents the intercept, which is always 1. And the second column represents a condition treatment with 0 for control samples and 1 for treatment samples. The xj values essentially act as indicator variables to capture the effect of the treatment condition on the log2 fold change beta I estimation for each gene. Now let's talk about the size factor and how it's calculated. Size factor estimation. Here's the step-by-step explanation of how DESeq2 estimates the size factor. Step 1. Create a pseudo-reference, sample row-wise geometric mean. Step 2, calculate the ratio of each sample to the reference, or the pseudo-reference. Step 3, calculate the normalization factor, which is the size factor for each sample. Step 4, calculate the normalized count values using the normalization factor. Imagine we have 3 samples, S1, S2, S3, and we have 4 genes, gene 1, gene 2, gene 3, and gene 4. The first step is to create a pseudo-reference for gene 1. The pseudo-reference for gene 1 is a square root 10 times 15 times 20, which is equal to 14.14. And the pseudo-reference for gene 2 will be in the same way, the square root of 100 times 120 times 110, equal to 115.47. And the pseudo-reference for gene 3 is the square root 50 times 60 times 55, which is equal to 57.45. And the pseudo-reference for gene 4 is the square root of 5 times 8 times 6, which gives 6.55. For the second step, we calculate the ratio of each sample to the reference. For gene 1, in S1, the ratio is 10 over the 14.14 that we obtained, which is equal to 0.71. And for G1 and S2, 15 over 14.14, which is equal to 1.06. For gene 1 in S3, the ratio is 20 over 14.142, which gives 1.41. Repeat the same calculation for all genes and samples. Step 3. We calculate the normalization factor, which is the size factor for each sample. The normalization factor for S1 is 0.71 times 1.06 times 1.41 to the power of 1 over 3, which gives 0.98. Normalization factor for S2 is 1 times 1 times 1 to the power of 1 over 3, which gives 1. And the normalization factor for S3, 0.95 times 0.95 times 0.96 to the power of 1 over 3, which gives 0.96. Step 4, we calculate the normalized count values using the normalization factor. Normalized count values are obtained by dividing the row counts by the corresponding normalization factor. Normalized count for gene 1 in S1 is 10 over 0.98, which gives 10.20. And for normalized count for gene 1 in S2 is 15 over 1, which is 15. And the normalized count for gene 1 in S3 is 20 over 0.96, which gives 20.83. Repeat the same calculation for all genes and samples. Now let's estimate the dispersion. Now that we have normalized the data, we move on to the dispersion estimation step. Dispersion refers to the variability or spread in the gene expression counts across different samples. DESeq2 estimates the dispersion values for each gene, considering both biological and technical variability. High dispersion values indicate higher variability, while low dispersion values suggest more consistent expression levels. Let's go through each step of dispersion estimate in DESeq2, with an example using hypothetical count data for 3 genes, gene A, gene B, and gene C, and 3 samples, S1, S2, and S3. In step 1, we estimate the mean and variance. In step 2, we account the gene-specific variability. Step 3, we account for the biological and technical variability. Step 4, we plot the dispersion, which is fitting the mean to the variance. And step 5, shrinkage estimation of dispersion. For the mean and variance estimation, we calculate the mean and variance for each gene across the samples. The mean for gene A equal to 10 plus 15 plus 20 over 3, which gives 15. And the variance for gene A is 10 minus 15 to the power of 2, plus 15 minus 15 to the power of 2, plus 20 minus 15 to the power of 2 over 2. Which gives 12.33, and we calculate the mean and variance for gene B and gene C2. The mean for gene B equal to 110, and the variance equal to 100. And the mean for gene C equal to 55, and the variance equal to 25. For step 2, the gene-specific variability. Next, we examine the distribution of row counts for each gene across all samples. We want to quantify how much the counts of each gene vary from sample to sample. For gene A, the counts, which are 10, 15, and 20, the variability, which is the variance, equal to 12.33 as calculated in step 1. And for gene B, the counts 100, 120, and 110, the variance equal to 100. And for gene C, the counts 50, 60, and 55, the variance equal to 25. In step 3, accounting for biological and technical variability. Now we use the dispersion values obtained in step 2 to model the gene-specific variability. The dispersion values will be incorporated into the negative binomial distribution. Gene A dispersion equal to 12.33, gene B dispersion equal to 100, and gene C dispersion equal to 25. Step 4. Fitting the mean to the variance. We can plot the dispersion values against the mean expression levels for all genes. Let's assume the following plot. When the mean was 15, the dispersion is 12.33. When the mean is 110, the dispersion is 100. And when the mean is 55, the dispersion is 25. And here we have the dispersion plot. A dispersion plot in DESeq2 is a graphical representation of how the variance, which is the dispersion of gene expression counts, relates to the mean expression level. This plot is essential for understanding the spread of data points and estimating the dispersion values used in statistical analysis. Let's break down the elements of dispersion plot. The black dots on the dispersion plot represent individual genes from our RNA-Seq data. Each dot represents a gene, and its position on the plot is determined by the mean expression level of that gene on the x-axis and the estimated dispersion value on the y-axis. For the blue dots, they are the summary of the dispersion values for genes on different bins on the mean expression. DESeq2 groups genes into bins based on their mean expression levels. The blue dots represent the average dispersion values for genes within each bin. These blue dots help visualize overall trend of dispersion as the mean expression changes, while the red line on the dispersion plot is a smooth trend line, often a smooth curve or line of the best fit, that shows the relationship between mean expression and dispersion. It helps visualize the trend in dispersion values as mean expression levels change. The shape of the red line provides insights into the relationship between the mean and variance across the expression range. And step 5, shrinkage estimation of dispersion. The dispersion estimates obtained from the dispersion plot may be subject to random variability, especially for genes with low counts or limited information. Shrinkage methods help stabilize and borrow information across genes to obtain more reliable and moderated dispersion estimates. This is particularly important for genes with low counts, as the estimated dispersion may be influenced by noisy data. And now, the calculation of test statistic. DESeq2 calculates a test statistic for each gene to quantify the magnitude of expression change and the variability in the data. In this example, we will use the whole test statistic to calculate it for each gene. For gene A, the log2 fold change S1 versus S2 equal log2 15 minus log2 10, which is 0.58. The standard error is the square root variance gene A over the normalized counts for S1 times the normalized count for S2, which is the square root of 12.33 over 10 times 15. Which is 0.14. The whole test equal to log2 fold change over the standard error, which is 4.14. For gene B, the log2 fold change, we do the same calculations, and we get the whole test, which is 4. We do the same for gene C, and the whole test statistic equal to 5.75. The whole test statistic measures the significance of the differential expression for each gene between the conditions being compared. For example, S1 versus S2. Higher values of the whole test statistic indicate more significant differences in gene expression. And now, statistical significance analysis. Firstly, we calculate the p-values and then the adjusted p-values. For the p-values, The p-values are then computed from the test statistics, representing the probability of observing such a difference in expression levels due to random chance. The p-values tell us whether the observed difference in expression is statistically significant or likely to occur by chance. A low p-value, typically below significance threshold 0.05, indicates that the observed difference in gene expression is unlikely to occur by chance alone, suggesting that there is a true biological difference between the conditions. The higher p-value indicates that there is no biological difference between the conditions. However, p-values are subject to the multiple testing problem, especially in high-throughput out data like RNA-Seq, where numerous genes are tested simultaneously. This can lead to an increased likelihood of false positive, where genes are falsely identified as differentially expressed when in reality they are not. To address the multiple testing problem and control the false discovery rate, DESeq2 calculates adjusted p-values using methods like the Benjamini-Hochberg procedure. So for the calculation of adjusted p-values, it takes into account the number of statistical tests performed and provides a more stringent measure of significance compared to the individual p-values. By controlling the FDR or false discovery rates, the adjusted p-values help to minimize the risk of false positives and provide a more reliable assessment of which genes are genuinely differentially expressed. At the end of the video, I'd like to mention that we perform differential gene expression analysis in R with DESeq2. I will leave the link of the video in the description below. Thanks for watching. I hope you enjoyed the video. If yes, then please don't forget to subscribe to our channel, hit the like button, share the video, and leave your comment under the comment section. Until next time, happy analyzing.